Good evening, everyone. We will maybe wait for another 2 to 3 minutes and then we will start with the class. Okay. So yeah, once again, good evening, everyone, and welcome to the week 4 live class session on the course on immunology. I am Srijan Sen from IIT Kharagpur, and today I will be taking the week 4 live class session. Right. So I'm sharing my slides with you all. I hope my screen is visible to you all. Yep. Good. Okay. So in today's class, we discussed a little bit of what I will be teaching today in last class. That's the generation of antibody diversity and things like that. So we will be starting with a quick revision of what we had went through throughout. And what I've done this time is basically I have kept the questions in between so that it's a lot more interactive, because what happens in the end, it comes all the questions together, and people might get confused in the end. They might forget the concepts and things like that. So I've tried to keep the questions in the middle, and I hope that helps really. Okay. So right, so today's class is about the generation of antibody diversity and TCR rearrangement. So we'll start with the immunoglobulin gene organization and VDJ recombination. We talked about this last class. So starting with the historical foundation, which deals with the two gene models that was proposed by in 1965 by Dreyer and Bennett's hypothesis, who proposed that the two separate genes, one for the V region and one codes for the C region, must somehow join at the DNA level to form a single continuous message. It's a radical at the time that's violated the prevailing one gene, one polypeptide principle. The second hypothesis being set up in 1976 by Tonegawa and Hozumi, which is a direct proof of this two gene model, who compared the restriction fragments from the embryonic DNA and compared it with the differentiated myeloma DNA using radio-labeled mRNA probe. So embryonic DNA, it has a V and C sequence on separate restriction enzymes, restriction fragments. The myeloma DNA has V and C sequence on the single fragment, so the DNA had been rearranged during differentiation. So Tonegama was awarded the Nobel Prize in 1987 for such a discovery. So for detecting the rearrangement, that's the southern blot logic. So you can see the intervening DNA sequence that are separated by large distances over here. So these are large restriction enzyme fragment that's detected. So the southern blot is differentiated, shows the differentiated fragment sizes because of the rearrangement that deletes the intervening DNA sequences and the restriction sites. So you can see the large fragment over here and some novel smaller fragments of around 2.3 um, kilobase pairs. Okay. Now, the human heavy chain locus in the chromosome 14 is basically involved in the germline configuration. So we have 51 segments of this variable region gene segments, followed by this DH, which has 27 segments, and then the constant region is separated into the different gene segment isotypes, starting from the 5 prime to the 3 prime region. So that's the multi-gene organization in the heavy chain locus. So what are the key features of it is that rearrangement requires two sequential joining events, the DH to the JH first, and then VH to DH JH. So the resulting VH DH JH unit, the altogether, the entire unit encodes the entire heavy chain variable region. The CH gene is arranged sequentially, which you can see from over here. How it goes? It goes, so there is arrangement of in the different ways, C mu, C delta, C gamma 3, C gamma 1, and over here it goes to epsilon, alpha. So this order matches the sequence in which the isotypes are expressed during the B cell response. So each V gene segment is preceded by a leader exon or the L exon encoding a signal peptide that is cleaved before the chain can be secreted. So the mouse heavy chain locus has around 134 VH segments, 13 DH, 4 segmental JH segments. Those are the key features of the heavy chain locus. So the light chain loci somehow looks like this, where you have 40 segments of this B kappa, 1 to 40, then you have the JK, then the CK. And the lambda locus that's present in chromosome 22, so that K locus is in chromosome 2. So that is V lambda 1 to V lambda 31, 31 functional genes. Then you have the downstream genes that are also present. So the key features of these light chain loci is that there is, for the K locus or the kappa locus, you have only one joining event, that is the V kappa to the J kappa, and any VK can combine with any of 5 JK, that's one pseudogene, and a single kappa segment has no kappa subtypes, right? So light chain rearrangement always occurs after heavy chain rearrangement is completed productively. And the lambda locus that's joins to one of the 4 functional J lambda segments. Each J lambda is paired with its own adjacent C lambda gene, that's lambda 1, 2, and 3. And J lambda 4 is a pseudo gene, yet its C lambda 4 partner is fully functional. These are the key features of the two loci. So the chromosomal locations of these genes is that the lambda light chain is present in human chromosome 22, and in mouse it is in number 16. The kappa light chain is in number 2 for humans and 6 for the mouse chromosome, and the heavy chain is in 14 and 12 for human and mouse respectively. Now there is a recombination signal sequences. We had studied about it, the RSS. So recombination signal sequences and the 12 by 23 joining rule. So one turn RSS is a 12 base pair special that flanks the V kappa, J lambda, and one side of the TH. So you see you have a V segment, you have an heptamer sequence, and then the 12 base pairs, that's one turn, and a nonamer, followed by the two-turn recombination signal sequence, that's a 23 base pair special, which flanks the J kappa, V lambda, VH, and the JH. So you have a J segment over there, followed by the heptamer. So you can see that the heptamer sequences are basically the same, as well as the nonamer sequences. And there is, in between the heptamer and the nonamer, you have a 23 base pair, that's a two-turn model. So the 12 by 23 rule says that a segment flanked by a 12 base pair one-turn special can join a segment flanked by a 23 base pair two-turn special. So one-turn RSS to a two-turn RSS is the productive pairing. So each RSS is the conserved palindromic heptamer plus the 80-rich monomer separated by conserved 12 base pair or a 23 base pair special. Right? Okay. So the 12 by 23 rule governs the correct joining. So one turn RSS, that is the 12 base pair special, can only join a two turn RSS, never two of the same type. So this rule ensures segments join in the correct order and type. So one turn V kappa joins the JK two turn, V lambda two turn joins the J lambda one turn. So the order is reversed between kappa and lambda. So VH and JH both carry two turn RSS. DH is flanked by one turn RSS on both the sides, and this forces D to join both V and J and prevents V from joining directly to the J. So what is the net effect that happens? So VL joins only the JL, never a VL, and VH, DH, and JH joins in the correct VDJ order. So that is what the 12 by 23 rule governs. So the VDJ recombinase machinery is mostly controlled by the recombination activating genes, which was identified in 1990 by Squats, Ottinger, and Baltimore. They act synergistically together to recognize the RSS, they synapse the two segments and introduce the DNA breaks. And then the lymphoid lineage is restricted, which is expressed only in developing the B and the T cells. Then you have the role of the terminal deoxynucleotide transferase, the TDT. It's a template independent DNA polymerase that adds N nucleotides at the coding joints. RAG1, RAG2, and TDT are the only lymphoid specific gene products that is required for the VDJ rearrangement. So the mechanism of this recombination starts with the RSS recognition and synapses, where the RAG1, RAG2, or the RAG2 recognize a one-turn and two-turn RSS and brings the two gene segments into proximity. That is governed by the 12 by 23 rule. Then there is a single strand DNA cleavage where the RAG1 or the RAG2 nick one strand at the border of each signal sequence and codes the sequence, followed by a hairpin formation and double strand break. The free OH, 3' OH attacks the opposite strand, forming a covalently sealed hairpin at the coding end and a blunt 5' phosphorylated signal end. The P nucleotide addition, that's asymmetric hairpin opening by an endonuclease that creates palindromic sequences at the coding ends. Then comes the role of TDT, where it adds the N nucleotide addition. So TDT adds up to 15 random nucleotides at the heavy chain coding ends, DJ and the VDJ joints. Lastly, it goes for a repair and ligation. Double strand break repairs enzymes, joins the coding ends, forms the coding joint, and the signal ends over there. So the signal joint, which is excised as a circle. So how it goes? First is the recognition, followed by the cleavage activity, hairpin formation, or the double strand break, and then You have the nucleotide trimming and addition. Lastly, repair and ligation. So coding joints, signal joints, and joining orientation. It says the two products of recombination, the coding joint, which is the functional VJ or the VDJ unit that falls between the coding sequences, and the signal joint that is formed between the two RSS. Then there is deletion joining, which occurs when segments are in the same transcriptional orientation, and the signal joint and the intervening DNA are excised as a circular DNA product and lost from the chromosome. Then is the inversional joining, which occurs when segments have opposite orientations, half of human weak segments. The intervening DNA, including the signal joint, is retained but inverted in place. So this is the different types of deletional joining, inversional joining, what you get. So what happens over here, you see for deletional joining, the V and J is oriented in the same direction. For an inversional joining, V and J is oriented in the opposite direction, right? Okay. Okay. So what happens before recombination? The V segment and the J segment each have their adjacent RSS, specifically of the 23 base pair RSS and 12 base pair RSS facing each other. The DNA between them is called the intervening DNA. You can see that. And after recombination, there is VJ coding joint. The V and the J segments are ligated directly together. This functional joint remains on the chromosome to be transcribed. And then you have the excised signal joint, where the two RSS elements are joined together to form a closed circular piece of DNA containing the intervening segments. Now this circular DNA fragment is lost from the chromosome and eventually degrades as the cell divides. Then there is inversional joining. So it occurs when the V and the J segments are oriented in opposite directions relative to each other on the chromosome. So before recombination, so because of the segments face the opposite ways, the RSS motifs are not facing each other directly in a linear fashion. So there has to be a recombination action to bring the 12 base pair and the 23 base pair RSS together, where the intervening DNA must form a coiled loop. Now this coiling twists the orientation of one of the segments. After recombination, the VJ coding joint, where the V and the J segments are successfully joined together on the chromosome to form a functional gene. And the retained intervening DNA, because of the looped geometry, is required for the alignment. The intervening DNA does not get deleted or lost. Instead, what happens, it flips. So it gets inverted and remains fully integrated within the chromosome, flanked by the joined RSS signal. Okay. So coming to the productive versus non-productive rearrangement. So junctional imprecision has a cost. Coding joint formation is imprecise. Unlike the precise signal joint, segments can be joined out of a triplet reading frame. So the non-productive rearrangement, where the reading frame is disrupted, there is premature stop codons and no functional protein. And productive rearrangement, where the reading frame is preserved, full length chain can be translated. So this is the consequences of B cell development. So cells attempt sequential rearrangements on alternative alleles if one is not productive, but at low success rates. Roughly 1 in 3 of various attempts, means fewer than 11% of the pre-B cells successfully mature and leaves the bone marrow. So then you have allelic exclusion, one cell, one specificity. So what is allelic exclusion? So B cells are diploid, having two copies of means having two copies of each chromosome. So the rule is that each mature B cell expresses immunoglobulin from one heavy chain allele and one light chain allele. So the purpose is to prevent a single B cell from being multi-specific, and at the same time it preserves the principle of clonal selection where one cell targets one specific antigen. So there is a feedback mechanism, that is the Yankopoulos and Alt model. So this model explains the step-by-step feedback loop that enforces single specificity expression. So there is heavy chain rearrangements. The cell undergoes a productive VH, DH, JH segment, a gene segment rearrangement on one particular allele, and this leads to the expression of the mu heavy chain protein, followed by a stop signal. So the presence of the protein signals the cells to shut down further rearrangements on the second heavy chain allele, and the same signal simultaneously initiates light chain gene rearrangement. Followed by there is a light chain joining where a productive light chain rearrangement occurs. So the new light chain pairs with the existing heavy chain to form a complete B cell receptor, that is the BCR. And finally, the shutdown mechanism where the formation of the complete BCR complex shuts down any further light chain rearrangement. This is supported by the transgenic mouse studies where a prearranged transgene blocks the endogenous heavy chain rearrangements. Okay, so based on this, I have a few MCQs you can try answering. So the first one says you can answer in the chat box. V gene segment in the human Ig heavy chain locus is flanked by two turn, 23 base pairs special RSS. So according to the 12 by 23 rule, it can recombine with a segment flanked by. Yes. So according to the 12 by 23 rule, it must pair with a segment that is flanked by one turn 12 base pair RSS, which is the DI segment in the heavy chain locus. So the correct answer is B. Okay. The next one says which enzyme pair is directly responsible for recognizing RSS synapsing gene segments and introducing the initial DNA breaks in the VDJ recombination. Yes, that is also correct. The RAG1 and the RAG2, which is the recombination activating genes. Good. Okay. The third one says pre-B cell rearranges its V kappa and J kappa segments out of the normal triplet reading frame, introducing a premature stop codon. This is best described as. So, what type of rearrangement is it? So maybe we can go one by one. So productive rearrangement, this occurs when the gene segments join in frame, allowing the synthesis of a complete functional immunoglobulin chain. Number two, which says the non-productive rearrangement occurs when the VDJ or the VJ segment joins out of frame. So this shifts the triplet reading frame of a genetic sequence. And as an outcome, the frame shift introduces a premature stop codon, preventing the synthesis of a functional immunoglobulin protein. So the correct answer is B. But we will go with the other two as well. So what is the class switched rearrangement? This process alters the heavy chain constant region, for example, from the IgM or the IgG. Later in the B cell development, it does not involve the V kappa or the VJ light chain segments. And somatically hypermutated rearrangement, this introduces point mutations into rearranged variable regions within the germinal centers to increase antibody affinity. So it is not responsible for the initial framing of the VJ joints in the pre-B cells. Okay. The next one says allelic exclusion of B cells ensures that. So allelic exclusion in B cells, what does it ensure? So simultaneous expression of both maternal and paternal alleles does not happen during the allelic exclusion. So A is incorrect. Then the choice of which parental allele, maternal or paternal, to express is random, not exclusively maternal. So C is also incorrect. And a single B cell expresses either kappa or lambda, never both. So D is incorrect. B is the correct option. So allelic exclusion. So this is a mechanism that ensures that only one of the two alleles for a gene is expressed in a single cell, while the other allele is silenced. This process is critical during the B cell development because it guarantees that a single mature B cell expresses a unique antigen receptor with single specificity. Right? Okay, good. The next one is deletional joining of V and J segments differs from inversional joining in that deletional joining. So we know that during VDJ recombination, the orientation of the gene segments determines whether the joining happens via deletion or inversion. So these are the only two ways. So deletional joining occurs when the V and the J segments are in the same transcriptional orientation. And when they are in the opposite transcriptional orientation, it is inversional. Right? The RAG1 and RAG2 complex brings the RSS together, cleaving the DNA. So this is happening for a deletional joining. So the coding segments are joined together on the chromosome, where the intervening DNA loop containing the signal joints is excised as a closed circular DNA molecule. And for inversional joining, the intervening DNA is not lost, instead it is flipped and written directly on the chromosome. So the correct answer is B. Good. Okay. Coming to the next part, that is the seven sources of antibody diversity. Okay. So the seven diversification mechanisms can be of all these types. So we don't need all of this. So right. So the source one is the multiple germline gene segments. So the germline contains an inventory of different VDJ gene segments. So the baseline variety represents the first and the simplest layer of antibody diversity before any cellular recombination or mutation happens. So you have the heavy chain, light chain, and the exact counts of these segments vary from person to person, from organism to organism. So this individual to individual difference is known as a allelic polymorphism. We had talked about it. For the combinatorial VDJ joining, so any VH can combine with any DH or any JH. So the overall diversity of antibodies is calculated by multiplying the heavy chain combinations by the light chain combinations, because of the heavy chain can pair with any light chain. So there can be like um, 8,000 8,262 possible VDJ combinations. There can be around 200 combinations of kappa, 120 combinations of lambda, and the total light chains is 200 plus 120, so 320 combinations. So leading to like 8,262 multiplied by 320, which is around 2,600. Sorry, 2 lakh. Uh, huge. Sorry, 26 lakhs 43,840, something like that. Okay. Okay. So yeah, that is what. So for human and mouse, you can get the individual numbers, and then you multiply the heavy chain with the total of the light chain, and you get those combinatorial associations. Okay. The next one is the junctional flexibility. So while the signal joint is always formed precisely, the coding joint is imprecise. The exact nucleotide position where the segments fuse can vary. This variability changes which codon spans the junction, altering the encoded amino acids at the point at the joint, even for the identical pair of gene segments. And then sequencing the same VK21 and JKappa1 coding joint in independent pre-B cell lines reveals different junctional sequences, while the corresponding signal joints are identical. Because of this variability falls within the CDR3, the loop that most often contacts the antigen directly is the junctional flexibility disproportionately shapes the antigen binding specificity. Coming to the fourth source, which is the p-nucleotide addition. It occurs during the hairpin resolution at the coding end. So the RAG-mediated cleavage seals the coding end into a hairpin loop. An endonuclease opens this hairpin, sometimes at an asymmetric position, leaving short single-stranded overhangs. The repair enzymes fill up this overhang using the existing strand of template, generating short palindromic sequences at the coding joint. The variation in exactly where the hairpin is cut is the variation that is generated in the coding joint sequence. The N-nucleotide addition is mostly catalyzed by the TDT. So TDT adds up to 15 nucleotides in a template independent fashion at the coding ends. And it occurs primarily at heavy chain joints, giving complete unit. So this was confirmed by the fibroblast transfection, and because of N addition occurs at the VDJ junctions, it localizes squarely within the CDR3 of the heavy chain, a major contributor to the heavy chain diversity. this is the P and the N nucleotide addition at the coding joint. So P nucleotides are templated nucleotides that form the palindromic sequences. It happens in 4 stages: hairpin formation, asymmetric cleavage, the flap extension, and the ligation. And the N nucleotide addition happens where the hairpin opens up, there is the TDT activity, strand pairing, and lastly the exonuclease activity and the repair. So where diversity localizes the CDR1, 2, and 3. So the sequence encoded by the V segment CDR1, the V segment CDR2, and at the junction this is the CDR3. Then you can check what are the things that are happening for these individual diversifications. Okay, so CDR3 is the hypervariable hotspot. CDR1 and 2 are encoded entirely within the germline V segment, so their variability comes from which V segment is used, plus later the somatic hypermutation. CDR3 sits precisely at the VJ or the VDJ junction, the epicenter of junctional flexibility. There is P addition and N addition. Because CDR3 usually makes the most extensive contact with antigen, concentrating multiple independent diversification mechanisms, that is highly efficient. It maximizes the functional diversity where it matters the most for antigen recognition. Then coming to the somatic hypermutation, which is source 6, it introduces point mutations into the already rearranged VJ, VDJ units, further diversifying a mature reporter after antigen exposure. Occurs only within the germinal centers, roughly a week after immunization with a T cell dependent antigen. It's targeted to around 1500 base pair window spanning the rearranged region. The frequency of around 10 to the power minus 3 per base pair generations, so at least a million fold higher than the spontaneous mutation rate. And mostly the nucleotides substitution occurs in a largely but not completely random fashion, so certain motifs and palindromic sequences are more susceptible. And mutations cluster within the CDRs of mature antigen selected B cells, where they're most likely to influence the antigen affinity. So affinity maturation, which was done by Berek and Milstein experiment. So the experimental design was somehow like this. So mice was immunized with a hapten carrier conjugate called the PFOX. Then the hybridomas were prepared from the spleen cells that were harvested at day 7, and after subsequent secondary tertiary immunizations, mRNA sequencing detected specific deviations from the known germline sequences. So the key findings were that in day 7, nearly all the early hybridomas used the germline genes with very few mutations. From day 14 onward, the overall mutation number increases. More hybridomas begin to switch to alternative V8 gene segments. And later in the secondary and the tertiary responses, mutations heavily clustered in the CDR1 and CDR2, and this leads to the progressive increase in antibody affinity for the FIFOX, which was characterized by lower dissociation constant. So the selection logic was that somatic mu hypermutation is random with respect to functional outcome. It can improve, worsen, or have no effect on the antigen affinity. The germinal center selection is not random. Somatic hypermutation plus selection is affinity maturation, and this is why antibody affinity for a given antigen rises measurably across primary, secondary, and tertiary immune responses. Coming to the last source, which is the combinatorial association of H and the L chains. So heavy and so the independent rearrangement where the heavy and the light chains rearrange separately in cells, allowing any rearranged heavy chain to pair with any rearranged light chain. So theoretical calculations often overestimate actual diversity because pairing efficiency varies, and usage is skewed for certain gene segments. But otherwise, what you see, we have talked about these numbers even before. Okay. Yeah, a few questions on these seven sources. The first one says, which of the following is not one of the seven recognized sources of antibody diversity? Yes, the class switch recombination. Right. N nucleotide addition catalyzed by the TDT differs from the P nucleotide addition in that N nucleotides are. So, what are these N nucleotides? Yes, so it's added in a template independent random fashion. That is B. Okay. Now, CDR3 typically shows the greatest sequence diversity among the three CDRs because it is, yes, so because it is located at the VJ or the VDJ junction where the junctional flexibility, P addition and N addition all converges. Right. Okay. Now, somatic hypermutation occurs at a rate of approximately 10 to the power minus 3 per base pair generation. Compared to the spontaneous mutation rate elsewhere in the genome, that is around 10 to the power minus 8 per base pair per generation, that would be how many fold higher, or would the rate be roughly the same? So, how do you do it? So the fold difference is equal to somatic hypermutation rate divided by background mutation rate. So the fold difference is what has been mentioned over here is 10 to the power minus 3 divided by 10 to the power minus 8, which is around 10 to the power, which is 10 to the power 5. That would be option C. So about 1 lakh fold higher. Yes? Okay. Next. Okay. Now, in the Berek-Milstein PFOX experiment, what change was observed in hybridomas isolated after Secondary and tertiary immunization compared to day 7. We observed what? Yeah. So more mutations clustered in CDR1 and 2 and higher antibody affinity for PFOX with lower KD value. Okay, fantastic. Now coming to the TCR gene rearrangement. So the T cell receptor has two lineages, alpha beta T cells and the gamma delta T cells. So what are these and how are they different? So the alpha beta T cells make up around 90 to 95% of the peripheral T cells. They recognize the peptide antigens presented on the MHC class 1 and 2 molecules, and they include CD4 plus and CD4 plus helper and CD8 plus cytotoxic subsets. And the gamma delta T cells, they represent a smaller population. Often they recognize antigen independent of the classical MHC presentation, and it's important at the epithelia and mucosal barriers. But they have a key similarity. Both the lineages assemble their receptor genes using the same recombination machinery as the immunoglobulins. And this process relies on the RAG1 and RAG1, RAG2 RSS within the 12 by 23 rule and the DSBR enzymes. Okay. Now this is the TCR versus IG locus organization. So here you see the details, the genetic architecture of the immunoglobulin and the T cell receptor loci. So the key important takeaways would be the light chain versus the heavy chain analogies, where the alpha gamma TCR loci resembles immunoglobulin light chains kappa and lambda because they lack the diversity segments and use simple VG joining. The beta and the delta TCR loci resembles the IG heavy chains because they contain the D segments and undergo VDJ joining. Then there is a nested locus. The TCR delta locus is uniquely nested entirely within the TCR alpha locus on chromosome 14. And the TCR alpha chain features an exceptionally large number of J segments, that is around 61. Okay. Yes, so these are some of the locus details. You can go through it once when I share the presentation. You can go through it. It's the same thing what is given over here. Right. So the shared machinery by the IG and the TCR recombination. So we have talked about it that both use the RAG1, RAG2 to recognize the RSS and catalyze the double strand breaks. So both follows the 12 by 23 rule for correct segment pairing. Both generate junctional diversity via imprecise coding joint formation. Both of them uses the TDT for N nucleotide addition, the TCR junctions, especially the CDR3, and they even show heavier use of in addition than the IG because TDT is highly active throughout the thymocyte development, and both are governed by the allelic exclusion. So the key differences being there is no somatic hypermutation, so the TCR genes are not hypermutated after antigen encounter. So the T cell affinity for peptide, that's MHC, is essentially fixed at the point of thymic selection. No class switching occurs, so TCR has no equivalent of isotype switching. So the C alpha, C beta are not swapped for alternative constant regions. And then there is heavy reliance on combinatorial plus junctional plus N addition diversity. Because two of the three post antigen refinement mechanisms are absent, T cells rely even more on maximizing the diversity before antigen exposure, especially via the N nucleotide addition at every junction. Sorry. Okay. Now, this is more or less a side by side comparison in a tabular form. So, what you see over here is the recombinase feature is there in the RAG1, in immunoglobulin and the TCR. You have both. It's the 12 by 23 rule governs both the immunoglobulin, the B cell, and the T cell. The junctional diversity happens for both of these. Somatic hypermutation is there for immunoglobulin, but not present in the TCR. The class isotype switching happens for B cell, but not for a T cell. Okay. Then you have the secreted form in the form of antibody, yes, but there is no so membrane bound only for a T cell, and then there is allelic exclusion, which is strict H and L, and for T cell it's strict for beta and relaxed for alpha. Now, the allelic exclusion at the TCR loci. So the TCR beta locus, which is strict allelic exclusion. So a productive TCR beta rearrangement pairs with an invariant pre-T alpha chain to form the pre-TCR with signals to suppress further beta locus rearrangement on the other allele, closely analogous to the heavy chain allelic exclusion. The TCR alpha locus is the exclusion is less strict, so both the alleles may attempt the V alpha, G alpha rearrangement, and the rearrangement can continue in reinitiating the secondary rearrangements until a cell is positively selected. And some mature T cells express the two different TCR alpha chains. So there is dual TCR alpha T cells that are recognized in minority phenomenon. Okay. So the T cell development and the order of the rearrangement. So yes. So first we have the DN or the double negative stage where the surface markers are CD4 negative and CD8 negative. So lacks both the co-receptors. So the TCR beta, gamma, and delta genes rearrangement begins here. So the TCR subunits rearrange first in this DN thymocytes. Then there is beta selection where it is a checkpoint for the productive TCR beta chain. So the newly formed TCR beta chain pairs with a surrogate pre-T alpha chain to form the pre-TCR. So this signals the cell to make a lineage choice between the gamma delta or the alpha beta T cell pathways, followed by the double positive stage where the surface markers it expresses both the co-receptors CD4 plus and CD8 plus. There is TCR alpha rearrangement. So TCR alpha rearranges only after a successful beta selection. Multiple rearrangement attempts are possible because there is no allelic exclusion pressure at this specific loci. Next, we have the positive or the negative selection. So this is a thymic selection process where the fully assembled alpha beta TCR is tested against self peptides MHC complex. The positive selection ensures that the TCR can recognize the self MHC molecules, and the negative selection eliminates the self reactive T cells to prevent autoimmunity. The last one is the single positive stage where the surface markers, either the CD4 plus or the CD8 plus, expresses only one of the co-receptors. The cells over here becomes a mature naive T cell and exits the thymus into the peripheral circulation. So the B cell selection and the lineage commitment. So the double negative DN thymocytes begins rearranging all of these in parallel. Then there is the checkpoint followed by the double positive so all the details are given over here. Okay. Okay, so for the double positive thymocytes, they now rearrange the TCR alpha. This results in the alpha beta TCR is tested for by the both positive and negative selection against the self peptide MHC complex. So why does immunoglobulin so why does immunoglobulin and TCR rearrangement stay lineage restricted? Okay. So despite using identical RAG1 and RAG2 RSS machinery, complete IG genes rearrangement occurs in the B cells and complete TCR gene rearrangement occurs only in the T cells. So the proposed mechanism is there is a regulatory sequence within the Ig kappa 3' enhancer normally prevents the V kappa J kappa joining in the T cell. So mutating this site allows kappa locus joining to the occur even in T cells, implicating lineage specific enhancer binding proteins as the gatekeepers. Similar accessibility based regulation is thought to restrict the TCR locus rearrangement to the T cell lineage. So, how do we estimate the TCR combinatorial diversity? So, you see, it's more of a calculation. So, the B chain has around 50 V beta, 2 D beta, and 13. So, multiplied by all of that, you get more than around 1000. So, 26 into 50 is like um, 1300 combinations. The alpha chain is around 70 into 61, so that should be like 4270 something. So the combinatorial alpha plus beta pairing multiples these further similar to the principle in the H plus L pairing. So because the TCR lacks somatic hypermutation, the junction diversity, especially the heavy N addition at both the VDJ beta junctions, becomes the dominant contributor to the CDR3 diversity, which is the most important thing in IG. So overall, estimates place potential alpha beta TCR diversity in a similar order of magnitude to the antibody repertoire, despite different mechanism wing. Now coming to the CDR loops and peptide MHC contact. So you see it's a TCR alpha beta heterodimer structure which is so the TCR alpha chain is represented on the upper side in light blue gray ribbons containing the beta A and the beta B sheets. The TCR beta chain is represented on the lower by the tan brown ribbons also containing the corresponding beta sheets. So the orientation is the top-down view which shows how the variable domains of the TCR face downward to scan the peptide MHC complex or the PMHC. Now the complementarity determining regions of the CDR loops. So the TCR uses 6 hypervariable loops, 3 from each chain, to recognize the antigen complex. And you see the CDR1 and the CDR2 loops, which is germline encoded. So these include the CDR1 alpha, CDR2 alpha, CDR1 beta, and the CDR2 beta. So they predominantly contact the outer alpha helixes of the MHC molecules itself, and because of they are germline encoded, they provide the baseline restriction that allows the TCRs to inherently recognize the MHC molecules. The CDR3 loops, so these include the CDR3 alpha and beta. They are positioned centrally and directly contact the bound antigenic peptide. Now these loops undergo VDJ junctional recombination, making them the most diversified regions. This allows them to specifically recognize unique peptide sequences. Now the antigenic peptide and the MHC interface. So the antigenic peptide is represented by the central string of purple spheres. It typically ranges from 8 to 11 amino acids for the MHC class 1, though class 2 peptides can be even longer than that. And the MHC helices, where the peptide sits in a cleft flanked by the two long parallel alpha helices, which acts as a cradle that exposes the peptide to the TCR. So, yeah, that's all. We can do some more questions, maybe. Okay, so it says, so the question says, which TCR gene loci use VDJ joining analogous to the immunoglobulin heavy chain rather than the VJ joining alone? So let's study about these two different polypeptide chains that undergoes the somatic recombination. So the VDJ joining by the beta and the delta gene loci, and the VJ by the alpha and the gamma. So yeah, beta and delta. That is correct. Okay. So the next one says the TCR delta locus is unusual among the antigen receptor loci because it is. So the T cell receptor delta chain gene segments are located entirely inside the T cell receptor alpha chain gene locus on the chromosome 14. So specifically the V delta, D delta and the J delta and the C delta segments are situated between the V and the J gene segments. So because of this nested arrangement, any productive rearrangement of the TCR alpha locus physically deletes the intervening TCR delta locus on that chromosome. So this mechanism ensures that a classic T cell expresses neither either a gamma delta receptor or an alpha beta receptor, but not both. So the right answer is physically nested within the TCR alpha locus. Okay. Which of the following diversification mechanisms is used by the Ig genes but not by the TCR genes? So you have to just get to the table that I showed. Yes, so the somatic hypermutation and class switching is used by IG but not by the TCR. That is option D. Okay. Formation, the next one, formation of the pre-TCR complex. That's the productive TCR beta chain plus the invariant pre-T alpha plus the CD3 during beta selection primarily functions to. Yes, so the correct option is B. So a productively rearranged TCR beta chain pairs with an invariant pre-T alpha chain and form the pre-TCR complex. So the signaling through this complex tells the cell that a functional beta chain has been successfully made. It shuts down the further rearrangements that the other TCR beta allele. So this ensures that the T cell expresses only one type of TCR beta chain. So right. The next one, a more simpler one. So allelic exclusion at the TCR alpha locus differs from that of the TCR beta locus in that. Yes, TCR alpha allows both alleles to attempt rearrangement, sometimes yielding T cells with two different TCR chains. So option B is correct. Coming to the next one. In which thymocyte developmental stage does TCR alpha gene rearrangement predominantly occur? So the TCR beta rearrangement happens during the first step, that is the double negative stage. So successful rearrangement leads to expression of the pre-TCR. Okay, and the TCR-A rearrangement, so once the cells becomes double positive, the proliferation stops and the TCR alpha gene rearrangement predominantly occurs. So this creates the final functional alpha beta TCR heterodimer, which is required for the positive and the negative selection. So double positive is correct. Okay. Now it says both IG and TCR loci use RAG1, RAG2 and identical these machineries, yet complete IG rearrangement occurs only in B cells and complete TCR rearrangement occurs in T cells. This lineage restriction is best explained by. So how do we explain it? So we know that differences in enzymes or the DNA sequences cannot explain the cell type specificity. The recombinase machinery can only bind to and cut DNA that is physically accessible. So B cells express specific transcription factors and enhancer binding proteins that open up the immunoglobulin loci, making them accessible to RAG. T cells express different factors that specifically open up for the TCR loci. So it is the lineage specific regulatory elements can be the enhancer binding proteins that control the locus accessibility to the shared recombinase machinery. Okay, the next one says because TCR genes lack somatic hypermutation, TCR receptor diversity relies especially heavily on. So the post-antigen affinity maturation in germinal centers is a feature that is unique to B cells, not the T cells. The MHC genes do not undergo ransom reassortment to create the TCR receptor diversity. They basically inherited codominantly. Uh, Even class switch recombination is a process that is used by B cells to switch the antibody types, so it does not apply to the TCRs. But unlike the B cell receptors, T cell receptors do not undergo somatic hypermutation or affinity maturation after meeting an antigen. So all TCR diversity is set before the T cell leaves the thymus, and this diversity is achieved through combinatorial VDJ recombination and junctional diversity, where the diversity includes the random addition of P and N nucleotides at the gene segment junctions, which increases the variety of the TCR repertoire. Yeah. Okay. The next one says which statement about TCR CDR loops and antigen contact is more accurate? So CDR1 and CDR2 loops are entirely encoded within the germline variable gene segment. So when the TCR binds to the MHC peptide complex. The CDR1 and 2 loops predominantly position themselves over the relatively conserved helices of the MHC molecule itself. The CDR3 loop spans the junctional region between the V, D, and the J segments. It undergoes extensive junctional diversity during this recombination. So, because of this central position, the CDR3 loop directly loops down to the interact primarily with the unique amino acid residues of the bound antigenic peptide. So, the correct answer is option C. Yes. And the last question, a T cell expresses productively rearranged TCR beta chain but has not yet rearranged a functional TCR alpha chain. This cell is most likely at which stage? So during the T cell development in the thymus, the T cell beta chain rearranges first. If the rearrangement is productive, the B chain pairs with the surrogate pre-T alpha chain to form the pre-TCR. Now, there is a phenotypic transition where the signaling through this pre-TCR triggers the cells to proliferate, arrest further beta chain rearrangement, and transition from the double negative stage to the double positive stage, expressing both the CD4 and CD8. Only after reaching the double positive stage does the thymocyte stop proliferating and initiate the rearrangement of its functional TCR alpha chain locus. So yeah, double positive thymocyte immediately after beta selection prior to completing the TCR alpha rearrangement. So option B is correct, right? Uh, Good. So if you have any questions, you can ask me. That's all from my part, the question answers, the lectures. If you have any questions, you can go ahead. No questions? Good enough then. Okay. So I think that's all. Again, we can meet in the next week on Friday at same time. Okay. So yeah, thank you all for joining. And this I will be sharing the lecture, and the recording will be uploaded in YouTube, which will be shared to you all by the NPTEL team. Thank you very much.